I am Jay Kim, and today I'll be talking about molecular biology. Cloning is using biotechnology to create a genetically identical replica. There are two major types of cloning: molecular cloning and reproductive cloning. Firstly, molecular cloning copies genes in DNAs by inserting them into plasmids, which are small DNA molecules that replicate the chromosomal DNA in bacteria. After the selected DNA fragment is inserted into the plasmid, which becomes a recombinant DNA, it is replicated into a bacterial host. As the bacteria divides and replicates by itself, the DNA fragment, which is the foreign DNA in the host DNA, is also replicated. Plasmids are ideal to conduct molecular cloning as they enable the selection of specific DNA fragments and the beneficial traits of organisms. These favorable DNA fragments are cut with restriction enzymes or restriction endonucleases. Restriction enzymes also cut DNA fragments so that they read the same forward on one strand and backwards on the other. Reproductive cloning is specifically used for multicellular organisms that often undergo sexual reproduction. Reproductive cloning is harder to achieve than molecular cloning since it requires two nuclei, each from an egg cell and a sperm cell. Therefore, there are two key materials required for reproductive cloning: a single diploid genetic complement and egg cytoplasm that creates the ideal environment for fertilization and early development. The nucleus from the egg cell is first removed in reproductive cloning. Then, the diploid genetic complement is inserted. As biotechnology develops, cells called embryonic stem cells are also introduced, which can develop into many other cells. Genetic engineering is a process to get desirable traits from organisms by using recombinant DNA to modify their DNAs. The outputs of genetic engineering are called genetically modified organisms, or GMOs. Two same organisms can be modified, or two organisms of different species can be modified, which are called transgenic genetic engineering. One approach in genetic engineering is called reverse genetics, which uses phenotypic effects of genetic sequencing to find out the function of a gene. Firstly, genome mapping indicates how the location of genes is identified in each chromosome. Genes are mapped with genetic markers, showing genetic linkages and interesting traits of each gene. These genetic maps have been developed over time into the form of physical maps, which also indicate the distance between genes. Genome mapping is effective to identify genes that cause illnesses and organisms with beneficial traits, becoming the database for future biotechnology such as preventing diseases. Genome sequencing is used to control and switch the DNA sequence of a genome. The very first genome sequencing were performed on small organisms and food like yeast and flies, from which scientists gain information about genes as a foundation. Now, genome sequencing is used to prevent the infectiousness and mutation risk of diseases, determine the effectiveness of drugs and medication, detect and trace down criminals, and even modify food to improve their taste and nutrition. Proteomes are proteins produced by cell types, such as tissues and cells. Proteomics is correlated with genomics, since proteins are encoded by mRNAs that are encoded by genes. Thus, proteomics can be used to prototype with and prove hypotheses of genes. When studying proteomics, Genomes remain constant, but the proteins inside an organism vary every time since they are produced in different tissues. An individual protein is called a biomarker, and a set of proteins with different expression levels is called a protein signature. Proteins can be used to detect diseases as well, as the condition of proteins in sweat, blood, or urine, or any other fluid or matter in the body is examined.